dance by the water beneath the Mexican sky. Drink some margaritas by a string of blue lights. Listen to the mariachi play at midnight. Are you with me? Are you with me?
Introduce the speaker today, Diego Orzáez, which is a research scientist at the IBMCP CSIC in Valencia. Diego did his PhD with Tony Granell, working in plant programmed cell death. And then he moved to the Netherlands to do a postdoc. He did there 2 postdocs, one working in the same topic from his thesis, and one working in the designing of plants as biofactories. Then he moved back to Valencia with a Ramón y Cajal contract in 2004, and he is, since 2009, a 10-year scientist at the IBMCP. Diego's contribution to the plant genomic and biotechnology, bioengineering field is amazing. He has published many, many papers in very important journals, and you can see in his CV, and his work right now focuses on molecular breeding, synthetic biology, genetic engineering, and the development of biotech tools. Actually, he has designed and created and developed the Golden Braid system for synthetic biology, which is a DNA molecular cloning system used worldwide in more than 100 labs. So you get a picture of the impact of his research. In addition, he is the co-promoter of 2 spin-off companies related to plant as biofactories and metabolic engineering. So as you can see, he's a great scientist, and I can tell you he is a great person. It's a pleasure to have you here, and we are looking forward to your talk.
Okay.
I don't know, this.
Okay. Thank you. Can you hear me with this? Can you hear me? Okay, thank you very much for the kind introduction. It's a pleasure to be here. Happy New Year to everyone. It's, as I said, I don't think there's any institute in Spain where I can see so many familiar faces, so I feel home, like very much Malaga. So thank you very much for the invitation, and thank you all for attending. Today I will. Actually, this morning I was looking at the presentation 5 years ago. Actually, there's no overlaps whatsoever. I was afraid did I overlap something, but I think everything is new. The title of my talk is about engineering gene circuits in plants, and I will try to entertain you during the next minutes. This is an outline of my talk. Basically, what I will start is with an introduction in which actually we do quite weird things, kind of uncommon things in bioengineering and so on. So I will try first to justify myself with you, my colleagues, and also as taxpayers, that what we are doing, it makes sense. I think it makes sense. So I will spend a few slides trying to justify myself, and then I will discuss 2 recent papers or works that we have been working recently. One is already published. That is a gene circuit for bioproduction. I will talk a little bit about autonomous bioluminescence in plants, because it's now our most favorite topic nowadays, and it will serve me as an introduction to explain you about our more recent development, that is the programming of bioluminescent plant sentinels, right? So this is the outline, and this is the most important slide. So afterwards we can, once you learn about this, we can finish the talk. I want to make sure that I contribute. I realize nowadays that when you are getting older, I'm getting older, I do not control most of the details, and actually the tribute is for the people, particularly the people in the lab, and most of the work I will tell you about is the work of PhD students like Camilo Calvache, Elena Garcia, Marta Rodriguez, and Victor Vázquez. And if there's someone in the lab that controls not almost everything is Marta Vázquez, who is on top of all of them, controlling all the details, and also tribute to her. And then we have a lot of collaborators from this work. I pinpointed the ones that are more related with this work, that are Karen Saskiyan, that I will talk about him. He's the developer of the first autonomous bioluminescent plants, and then other people in the Jose Antonio Doros in the IBMCP. And I will talk a little bit, because we use Gemini viruses as tools, and here are the masters of Gemini virus. We don't have any idea about Gemini viruses, but so we only learn, we use them as a tools, and we have a very nice collaboration with the people here, and also with Rosa Lozano, who teaches about everything about these tools. Okay, let's try to justify myself. At the end, the underlying question that we try to respond is that if we can really engineer plants, right? If we can make genetically engineer plants from a rational point of view. And this is a difficult question, and I don't know the answer yet. But plants are especially interesting organisms to try to engineer for two reasons. Why? Because from my point of view, the future of humankind depends in our ability to really engineer plants, because they are our source of most of the materials and food, and so definitely we need to learn, if we want to survive as a humankind in the future. We need to learn how how can we deal with this. And second, because multicellular plants are multicellular organisms, and but we don't have the ethical considerations that we have with if we want to engineer, for instance, mammalians and so on. If you want to do autofluorescent, autoluminescent mammalians, so like ET, probably we will deal with many problems, ethical problems. But with plants, we can learn which are the constraints for engineering without that big ethical constraints, right? So this is the point. I think most current traits that we are using now for engineering, for new traits, are coming from wide alleles or from random mutagenesis. We have a few synthetic traits from GMOs. I calculated things with the help of ChatGPT that nowadays between 10 and 20% of our caloric intake comes from GM crops. Consider that we are eating also animals that are fed with GM crops. So actually, we use very much of this biotech, but most of those are very hardwired traits, are not programmable traits, right? So our question is, can we generate new valuable traits for the future that can we really create programmable traits? And what is what we understand as programmable traits? Programmable traits are those whose behavior is explicitly designed rather than fixed, and can be tuned, combined, or conditionally activated by engineering genetic logic control. And I asked also ChatGPT to give me some differences between classical traits and programmable traits. And here you see some of them. The classical traits that we are using normally are always on. A programmable trait is one that is conditionally activated. And here you can see a number of characteristics, but the one that I'm most interested in is how to do them, how to produce those programmable traits. And one of the clear things is that classical traits, herbicide resistance, insect resistance, are single allele traits, where here you need to create modular genetic circuits in order to do those programmable traits, right? So, and what are those synthetic circuits that we want to use or learn to use in order to do those programmable traits, to create those programmable traits? So this is a little bit the basic of our lab, of what we do in our lab, right? Is to create these synthetic circuits that are rational design groups of genes, of genetic elements, promoters, regulators, coding sequences, that we assemble in order to process inputs and produce predictable outputs in living cells, right? So this is the basic, as I could say, the basic idea in our lab is to create those synthetic circuits that basically I learned that they have 3 elements, if we would want to, that standardized elements, right? One is a sensor, a processor, an actuator. This is the basic idea of what a synthetic gene circuit is. And a sensor, there are different types of genetic circuits, but the ones we use the most are transcriptional synthetic circuits. And transcriptional circuits are those that are composed by these 3 elements, sensors, processors, and actuators. And I will try now to define which we understand, how we understand each of these elements, right? And this is something very useful for us when we work in the lab, because this allows us to exchange information among the different people in the lab or with other colleagues. For us, a sensor is an element, a genetic element, that inputs a not transcriptional signal and outputs a transcriptional signal, right? This is what we understand as an input. A processor is a genetic element that inputs a transcriptional signal and outputs also a transcriptional signal of a different type. For instance, an amplifier or an integrator of different signals and so on. And then finally, an actuator, that is our enzymes, our biosynthetic pathways and so on, are elements that input a transcriptional signal and output a non-transcriptional signal, right? Like an enzyme and so on. So this is basically most of my talk will follow this scheme of sensor, processor, and actuator, right? So the main question we have in our lab, and I don't know the answer yet, is can we create robust new-to-nature traits by programming plant genomes with this kind of synthetic gene circuits, right? And it's something whose the answer we don't know, because we don't know to what extent we can make this in a robust way in plants. So long time ago, we started to approach this problem by using standardized hierarchical genetic elements. So the idea is that we start with basic DNA parts that combine in transcriptional units, that we combine this in multi-gene elements, functional sensors, processors, and actuators, and then at the end we put them together and create a gene circuit, right? This is the basic idea. And we have been for many years producing many of those elements, right? That are supposed to be, we try to make them in a standardized modular way. So we have, you mentioned about this golden braid. In our lab, we have more than 4,000 golden braid parts that we have from basic parts, promoters, CDS, terminators, to higher hierarchical level parts, transcriptional units, sensors, processors, actuators, and finally, complete gene circuits, right? And this is made in a way that are very easy to combine in kind of Lego, can be combined in a standardized way, kind of Lego pieces. So this is the trick of synthetic biology. These are the games or the rules of the game. So the idea is to use these standardized parts to learn how to build those new instructions, new programs in plants, and learn to what extent we are able to create new functions that are new to nature. And the other tools, or the additional rules of the game is we try to do this, as I said, using standard parts that can be exchanged every time, and to use the same parts and exchange the parts between different labs and different groups and different students. And we have we try to do it by do this as fast as possible using fast prototyping, using what is called in synthetic biology design, build, test, learn cycle. So basically, you don't have a final goal. What you normally do is to continue. It's like in the computer science, you have version 1.0, 2.0 exactly. You never arrive to the final goal, but you continue developing new cycles of development, trying to do an incremental development, right? So we have to think on, and you will see this later on, we have to think of very fast as possible ways of prototyping in order to reach the final goal. And our crop, the crop we normally focus, or the plant we normally focus is Nicotiana bentamiana. Nicotiana bentamiana for two reasons. First, because it's very easy to prototype those circuits thanks to transient expression, because as you know, we can very easily test the new circuits in transient expression using Nicotiana bentamiana agroinfiltration. And second, because Nicotiana bentamiana is already a chassis for bioproduction. Many companies, including some of the spin-offs we work with, they use Nicotiana bentamiana and transient massive agroinfiltration for production of recombinant proteins, metabolites, and so on. So every circuit that we create, we can translate it directly in a plant that is already in the industry as the standard for bioproduction, right? So this is the process. And having said that, I'm going to try to talk you about the first case. So after having developed all these different basic elements, promoters, circuits, and so on, what we have to do is to put them together and make something that, a conditional trait that really makes sense in the real world, right? So that's what the first case I'm gonna introduce you is a circuit that we developed a short time ago for conditional recombinant protein production in Nicotiana bentamiana, right? And the story about this is that when we want to use plants for bioproduction, the constitutive expression is a problem because you are taking resources from the plant all the time to produce your protein or your metabolite of interest, and this can be a problem because it interacts with the homeostasis of the plant. So the idea thing is to have a spatiotemporal control of the production, right? So our problem is the trade-off between tight control and high expression levels. And the solution we propose is amplify gene dosage using genome-integrated viral replicons. So our circuit, so what we want to produce is a conditional amplification of a foreign gene expression in response to an agronomically compatible signal. And this is the work, the PhD thesis of Elena Garcia, and basically what she devised, the circuit she developed is a circuit that contains a sensor that is a copper sensor. Copper is a widely used agrochemical, so the idea is to develop a copper sensor, a processor that is amplifying the signal using the Gemini viral replication machinery, and actuator that is actually the production of the final protein. So it's following the protein of interest, following the pathway or the context, the framework of these synthetic gene circuits, right? So Elena likes to do these analogies with the input is the singer, the sensor is the microphone, the amplifier is the Gemini viral replication machinery, and the loudspeaker would be the recombinant protein. So it's the analogy with a normal electronic circuit. And the system is called Cube because it's copper, and the Gemini virus processor is coming from BYD Gemini virus, right? So let's talk a little bit about the sensor. She developed time ago. We thought which kind of input, which is agronomically compatible, we could use in order to create the circuit. And she immediately thought about copper. This is her father, and she used to treat copper. So copper is something that you can use it's cheap, and you can use in crops, even in open field, relatively cheap. So what there was already described a CUP2 system that is a copper sensor that is transcription factor that responds to copper and and binds a region in a promoter, and can activate the expression of a certain protein. In this case, the rationale is to activate the expression in response to copper to a rep-A protein that is responsible of the activation of a Gemini viral system. So this would be the sensor part. Then, in order to produce the amplifier processor, what the idea, and here we rely on the experts, is to take advantage of the Gemini viral replicative machinery. As you all probably know, well, you know a lot, much more than me about Gemini virus, but from the biotechnological point of view, what is interesting for us is that you can integrate a replicon based on this Gemini virus in the genome simply by linking two lead regions and introducing a seed region, and with the presence of the rep protein, you can induce the circularization of a replicon, and that it will be amplified by a rolling circle replication, and you can maintain at the same time in the genome the original copy of the same structure. And this, at the end, is an amplification of the signal. So in that way, what you can do in the plant is to amplify the gene dosage of the recombinant of the protein of interest, right? So, and then the actuator, in this case, it would be a model actuator that is simply a fluorescent protein to learn how it works, right? So, as I said, we use Nicotiana. The idea is to integrate everything, all this machinery within Nicotiana ventamiana, because the standard chassis for plant bioproduction, the normal use, or the standard of the industry, is to use massive agroinfiltration in order to produce recombinant proteins. So you have big pools of agrobacterium, and you infiltrate large amounts of Nicotiana ventamiana, many plants in these reservoirs of agrobacterium, and this is technologically very expensive as a system, because at the end it's very demanding, because at the end you are working with two biological systems, with agrobacterium and with the plant, and this is really cost, implies high costs. And the alternative, we think that an ideal alternative is that instead of using these two organisms, you could have the same output, but simply deploying or activating the viral system, the amplification of the viral system, using a simple spray with, in this case, copper, right? So, here are a few details. This took a lot of time for optimization, and we do all the optimization via transient expression, and then once it's optimized transiently, we go to the stable plants, right? And it took us a while to optimize the Gemini viral system. It turned out that what Elena did is to use what we call the Geminino configuration of the vector, and it's a replicon of structure of the Gemini virus, that it has a a strategic configuration in which the promoter you see is downstream of the CDS, right? And here you have half of an intron. So to make a long story short, you can see here that the presence of rep, what it produces is the recircularization of the replicon, and then you have the promoter in front of the CDS. This doesn't happen in the linear structure. So in these conditions, only upon circularization in which you have gene expression, right? And this is important because one of the problems that we face when we want to integrate viral replicons in plants is leak expression. So as soon as you have some leak expression, this boosts the production of the replicon, and it spoils the whole experiment, because you only want to have this boost of expression when you spray with the inducer, right? So the integrated machinery of the amplification has to be very tightly controlled. So one of the ways to control this is by the configuration of the cube of the vector of the amplification system. And so once it's circularized and transcribed, the lead is within an intron, and then it's spliced out, and then you can have production. And this transiently works very well. This in the presence of the rep. You see a lot of GFP, nothing in the absence of the rep, so it's quite fine. But then still we have some leakiness, because the fact the copper inducer induction, the chemical induction, is always a little bit leaky, and this can ruin also your system. So we had to add to engineer an additional break to the system based on a recombinase. Look, instead, this is the activation of rep-rep-A that is the key for the amplification of the Gemini viral system, right? But this rep-rep-A is not directly activated by the copper factor with this promoter, but instead, in addition, we have what we call a break system, in which we have a terminator linked by recombination sites, right? What we activate is not, well, on top of activating with copper the rep-rep-A, we activate also a recombinase. The recombinase, when it's activated, removes this break, this terminator, and the transcription can go. And this has become, in all the optimization experiments that we do in DBTL cycles, it turned out that it was crucial in order to be able to produce plants. But otherwise, you cannot produce transgenic plants, because they are the system starts to work, the engine starts, and then you cannot recover transgenic plants. So this was crucial. This additional break was crucial in order to get. So after all this work, what we did is a system in which the copper activation, the copper transcription factor, activates both the recombinase and the rep-rep-A. And there's this loop with a double activation, and using a particular recombinase that is a flipase, we had a very good dynamic range with very low basal expression systems. And this is what finally Elena chose as system for bringing into stable plants. So the process is always lots of optimization steps by transient expression. Once we believe we are in a good position, then we go to stable plants. And the stable plants normally are rather complex to do, because as you can see, these are all the elements that require that had to be introduced in the transgenic plant to make it work. That contains the rep-rep-A with the brake system and the promoter that is regulated by copper. Then the recombinase itself regulated by copper. The transcription factor that activates all, that orchestrates all the system. A resistor, a marker, and then in another vector, all the processor part, that is the amplification part, with the Gemini viral vector, or the amplifier, right? So we have, structurally, we also separate our elements as sensor, processors, and actuators, right, in different vectors. But at the end, it works. That's the nice part, that it works. You go to T0 lines, and you can see with water and with copper leaf disc, you can see that actually it works. This is something probably I will discuss at the end. It's not 100%. Not all the transgenic lines you produce have the same behavior, but you can isolate some plants that have the behavior you expect. For instance, this like 3 in copper or in water, line 4 in copper or in water. And you can see that is a nice activation in response to copper, and you can see GFP expression in response to copper. The nice thing, well, to make a long story short, we tried different ways of activation. Spraying, as you can see, with 5 millimolar of copper sulfate, and waiting 4 hours, and you can see clearly the activation of GFP. With hydroponics, that also using growing in hydroponic condition and adding copper, you can get the activation, and this is very nice to see that the levels were quite high because of the amplification, up to 2.6% of the total soluble protein in the plant was. That is, there are systems that get higher, but this is pretty interesting, and it's quite clean. This is the plant with water, and this is as soon as you add copper sulfate. So it was quite clean. But what we found out, it was a kind of surprise, is that we could also, and this was probably one of the most interesting outputs, we could also activate it post-harvesting. So you can grow the plant, you remove the leaves. This operationally, it's very nice because you can grow the plant to maturity, you take out the leaves, you leave it in 5 minutes, the leaves submerged in copper, and then you leave them for 3 days in water, in normal conditions, and then you see also the activation, right? With water, but after copper activation, only with 5 minutes incubation with copper. This is, from an operative point of view, is very interesting because it allows us to grow the plants and you don't need to spray or anything like this. You can harvest, incubate, and produce the plants 1 hour are harvested to make them produce for a few days, right? So this is about GFP, but actually we don't want to produce GFP, right? We want to produce something that is more interesting. But once you have established the system, the nice thing is that, and I'll give you an example with a nanobody, but we have some other examples. You have already all the elements, and this is the nice thing about creating modular elements, right? You have the whole system, but then you can just change the GFP by a heavy chain antibody, a single chain antibody. And in that case, you start to recover plants and also use the same system for the production of recombinant antibody. This is in this case, it's anti-SARS-CoV-2 antibody. And well, I'm not going into details, but you see that the system works exactly the same without and with copper. Without and with copper, you can recover lines that post-harvest, you simply take it out and produce. And these are plants producing the antibody, uncontrolled plants without copper, or no, this is actually wild-type plants, and you can see this is an ELISA that they have the antibodies functional, and you can produce also. In this case, the levels were not that high, 0.45 milligrams per kilogram, but yeah, it's more than what you can expect definitely with a 35S promoter. So this is this story. So I hope I convince you that to a certain extent you can design a circuit and create a new trait that is new to nature, but it takes a lot of optimization, but you can then recycle it in order to produce different products, right? So the takeaway message is that the cube circuit allows the precise expression of recombinant proteins upon copper switch, and it can be activated easily with a spray, with watering, and what is most interesting, ex vivo with post-harvesting incubation of leaves, right? So this is the story. I hope you like it. And now my slides come to black because I'm gonna change gears, and the next slides will be all of them. Sorry? Okay. Because the next case I want to tell you is about sentinel plants. Oh, it's too much. Just otherwise, here is perfect. And now I think it will be nicer to see it with a black background. So this is not about bioproduction, but about sensing, right? And again, we have the problem is the need for affordable specific detection of biotic or abiotic stress. And what we propose is to create circuits that allow us to make plants work as plant sentinels in intercrop system, right? You have a crop, and then you have some plants that, this is the idea, right? That you intercrop, and they can serve you as a sentinel, right? And the programmable trait that we would like to produce in this case are specific stimulus-dependent activation of remotely activated outputs. And in this case, our output is autonomous luminescence in response to a defined stress. And in this case, in our case, a stress is a virus. This is sometimes use similar things in agriculture, like the sentinels. And here you see a tribute to the sentinel soldier. That's what in the literature normally you see as described as a sentinel plant or a plant sentinel. And for this, I need to introduce you something that we like very much, we enjoy it a lot because it's, from my point of view, very cool, is that it's plant the autobioluminescence pathway, the fungal and autobioluminescence pathway that the group of Karen Saskilisian, who is now in the Imperial College, before it was at the Russian Academy of Science, introduced in plants in order to produce bioluminescent plants. This is a pathway that starts, is a form from bioluminescent fungi. This pathway starts with caffeic acid, so it's something that is common in plants. So we can use plants or the pool of caffeic acid in plants in order to feed the pathway. This pathway is a circular pathway. Start with an histidine synthase that produces histidine. Then there's the enzyme H3H produces luciferin, that is this 3-hydroxyhistidine. And then there's an enzyme that they call LUTH that is the actual luciferin that produces photons of 525 nanometers. And then the whole system can be recycled with CPH, this other enzyme, and produces again caffeic acid. Histidine synthase, this is the key enzyme in the pathway. Normally, the fungal one needs to be activated post-transcriptionally with another enzyme that is called MPGA. And the presence of MPGA boosts the production of bioluminescence. It's not strictly required because there's certain activity in plants, but if you add MGPA, you boost the autoluminescence. And besides that, let me show you here, also to understand the next slides, that there's also, we published with the group of the Karen Saskiyan that there are certain enzymes in plants that can also activate do this pathway, are independent on MPGA and are smaller because histidine synthase is a very large enzyme. There are some plant native enzymes that can produce the same pathway that is this very specific step passing from caffeic acid into histidine. When you translate, when you put these 4 enzymes in plants, you can produce these beautiful pictures of bioluminescent plants. This is Marta Vázquez showing the Nicotina ventamiana with all the pathway. It's very beautiful, but it's not only very beautiful, very nice and very cool, it's also that we think it's very useful for our purposes. Our purposes in two senses, for 2 reasons. First, because it allows us it helps us to do this fast prototyping that I was telling you before. If we want to advance, there's no final goal. You need to go through DBTL cycles and try to understand and fine-tune the requirements to have a good cycle, a good circuit. And in that case, the output is very easy to record. And you can do agroinfiltration, transient expression, and get, I will tell you now, you get plenty of data, more than any other system that I've used before. And also, because allow us for remote diagnosis. Light has very low zero background in plants because plants do not produce light. Fluorescence, for instance, have background because you have autofluorescence, but in the dark, there's absolutely zero background in the case of plants. So this is very nice. And the interesting thing is that you can control, you can use it not only for the cellular level as we use in fluorescence, but you can go up into the organ level and also on the organism level, and even within into the ecological level, into the population level, as I will show you. So this is the way to use it as a reporter. You can introduce the whole pathway in the plant or in your circuit, and you connect, you plug your circuit of interest, because this will be the actuator in our concept of what is a gene circuit, and you can plug this actuator with any other processor and so on, using the promoter of either the histidine synthase or the LUTH, with any of these 2 enzymes of the pathway. Each one has a different advantages, right? We like to plug all our circuits in speed in synthase because it gives us a more linear output. There's no saturation whatsoever. It's very much similar to what we get with regular luciferase systems, the Firefly luciferase. But it's also, sorry, this one. But it's also very interesting to use LUT because in the case of LUT, the background is absolutely zero, right? I don't know if you follow me, but the idea is you have the actuator is the whole pathway, and then this will serve you as an output of any other circuit that you link to this actuator, right? So that's the basic idea. And, well, we have established a whole system to use this bioluminescence as output in our optimization system. And I told you before about copper sensors and so on. Now we recapitulated all our elements using this new system, and it's great because you produce a lot of data. You can produce because this is a living organism, so you don't destroy. You don't destroy your material, so you can do time course analysis. You simply leave them, leave this, for instance, leave them in your 96 well plates, and they produce data for 4 or 5 days, and you can see the dynamics and so on. And you don't use to add luciferase, as in the case of Firefly luciferase. You don't need to use the substrate because the substrate is caffeic acid, so it's in the plant. So it's very nice. But what is more even nicer from our point of view is that it allows you to record outputs at the organism level, at the organ level, and the organism level, and which has set up the whole lab to be able to detect. We have several setups in the lab to do this kind of detection, right? And I'm going to give you just a few examples to illustrate this. One of the things we did is to produce recombinant viruses that they contain one of these enzymes of the pathway. So what we plug the movement of a recombinant virus to the pathway. And the way to do this is in the genome of the virus, you introduce, instead of any gene of interest, that is something that normally you do with recombinant virus, you introduce the luciferase, right, or the speed in synthase. And in that case, and we have all the setup. By the way, it's quite intense the output. So you can record everything simply with cameras that we have connected with the software and so on. With a normal reflex camera, you can have all the data you require. And here you see, just with the camera, that you can trace the movement of the expansion of the viruses, right, through the plant, but also at the organism level, you can just put your plant, inoculate the virus, and the plants light up, and you can start see how they move, simply taking pictures at different times. And we have adapted also software for being able to record and quantify the levels of activation. This is the work of Victor Vázquez, who is a master of all these softwares. And we can, at every point, we can isolate images, quantify those images, and have very easily, almost automatically, a time course of what is going on in terms of expression of the virus and in different positions, right? So it's quite cool. So once we have established this, then we went to the production of circuits, and that is circuits for sentineling, right? So the basic idea is, can we use this system in order to warn us of what's going on in the plant in terms of a certain stress? And we have focused, for the time being, in virus detection, because what I showed you before is a recombinant virus. This is a trick, right? This is not real. But this is what we want to produce are the plant to switch on the light or to change the color when they detect the presence of a certain virus. This is a real sentinel, right? And the idea is to use it as intercropping in different with other crops. So we are trying to develop two different sensors, two different sentinels, one for detecting specific strains of Gemini viruses, and other for detecting potiviruses infections. I'm going to focus in this one, right, because it's the more advanced and also in the sake of time, and but we can discuss about the problems we're having also with the Gemini virus system. But this is a nice one, I think. I'm trying to convince you, because in this case, we don't want only that the plant switches on when detecting a virus. This is quite trivial. We would like the plant to change color once it detects the viruses. So it's normally, this is like a detector system, fire detector system. Normally, you need a proof that the system is on in the basic situation. Otherwise, you don't know if it doesn't respond simply because it's not working. So that's what we wanted to engineer. So this is the equivalent of this system, this pilot informing you about the status of the system, and the system is working. So the ideal thing is that you have stable the pilot in plant that is emitting light constantly at a low level, and then if you see the alarm, then it switches on and produces a different color, and it switches and produces more light, right? That's the idea. So that's what we try to engineer using in this case, it's not a transcriptional circuit, it's a post-transcriptional circuit for potivirus sentinel detection. And in this case, the idea is that the plant emits in yellow, and then when it's on, emits in green, right? How do we do this? The trick for doing this is that we take advantage of bioluminescence resonance energy transfer, BRET. Is the ability of this enzyme, if you fuse it to a fluorescent protein, it transfer the energy, and then there's a switch in the spectrum of emission, right? And you can detect this easily with a spectrophotometer or using filters, right? So first of all, so the idea to use as a spoiler is that tobacco H viruses and other potiviruses all of them have a specific protease because they are produced as a polyprotein, and there are specific proteases that process this polyprotein in a specific cutting cleavage site, right? So the idea is to engineer a cleavage site here. So when the virus arrives, it cleaves and this produces a change in the spectrum. So the first thing, there's a lot of optimizing here. I'm going to go fast, but the first step is to do partners of bread and to optimize bread partners to obtain an efficient change in color. I'm colorblind, so I told you before I trust the people in the lab, but in this case I'm completely, I'm on their hands. Of course you can quantify, right? But I hope you can see that there's certain partners which you can see a change in color, particularly if we use a filter. This is the original one that it's in green, I hope. And then there are certain bread partners that switch the output color into yellow-red. I hope you can see it. I don't see anything, but I trust you can see it. I trust this because there's also a shift in the spectrum that you can also measure using EVs. That is an equipment that have a panel of filters in order to do this, right? So we initially rely very much on this equipment, right? And yeah, and you also can use confocal in order to see that this is working. Anyway, so once we establish a good partner, and in this case, the DS-RED is a good partner, then we have to see if the protease of the potivirus, in this case we use tobacco H virus as our enemy, if we were able to use this protease, and the presence of this protease can change the spectrum, right? And actually, I hope you can see that in the presence of the protease, there's a change in color. So actually, the system is working and is processing and producing the change in the color. So the next step was not only not to use the protease, but also to agroinfiltrate the virus itself, the infective clone of the virus itself. To see it was sufficient in order to do the change in color. And again, I hope you can see that in the case of the presence of TEV, you can return into yellow, into green. Is that the case? You can see it in the green, sorry. And not in the case of another potivirus that has a different cleavage site, has also a polyprotein, also a protease, but a different cleavage site. So it's viral specific, right? So you are specific of the target that you are using, right? So I hope you can see it here. We also created a sentinel for the other potivirus, so we show that you can use it to detect several potivirus, and well, this can be detected specifically, right? So, well, and the next step, this is transient. The next step is to create the sentinel, so put all the machinery stably in the plant and create plant cells, plants that in the background, they are emitting in orange. Is that right? In orange. And in the presence of the protease or the virus, it changes the color, right? And I can convince you that this is the case. This is the background luminescence, and then in the presence of the TEF, there's this change, both if you use the protease or if you use the virus itself. And well, this is in this case our circuit, and this is the whole pathway is integrated in the plant, and then what it comes, the input is the potivirus itself that produces the cleavage. And you can monitor this in the stable plants. This is a plant infected with the with the non-specific potivirus, and this is with the TEV virus, and you can see during the time that that there's an activation, right? And it's not only that you see the switch on of the plant. These are pictures taken only with a plain camera, with a reflex camera. The nice thing is that you can also see in those plants that there's the DS red. One of the funny thing is that this complex normally is located in a microsomal fraction. It's associated with membranes, and in the presence of TEF, it's not only that it starts changing the color and growing and glowing more specifically, but it also that changes the mobility because it moves from the microsomal fraction into the nucleocytoplasmic fraction. So you are releasing and you are able to detect the change also of DS red in this case, right? So we can see a video in which you record, this is like the switch on, right? And in the case of TEV, but not in the case of TMEMV, you can see how the plant is switching on. But the difference is that before it was a transgenic virus, but here is the wild type virus that is infecting the plant, right? So, and this is specific, not only on potivirus, but any other infection of the virus that doesn't switch on the plant and does not produce the change in color. And we have seen this many times. And then the final experiment that we did, we submitted this to a journal, and they asked us for a additional experiment more on the population. So we are doing also the small experiments at the small population levels. This is our setup. You have, what we did is an intercropping of sentinel plants in between tomato plants. And we did a experimental infection with PVX, that is unrelated virus, and with different doses of the target virus, of the tobacco H viruses, and see what happened with the time, right? With the different inoculation with different doses of increased doses of the virus. So this is what happened. This is a sentinel picture. This is what happened with day 0, day 2, and as you see, you can start to see in the high doses of the virus of the target virus that this is switched on, and as you see after day 6 and day 8, you can see that the plants, the sentinel plants, are switched on. I have to say that we were very lucky because there's not only a change in color, but also a change of intensity due to the release of the bred partner, right? So this helps us also for the detection. So you can monitor this, and you can see how some plants are not activating, but you can monitor quantitatively both in beam maps and also measuring the levels of luminescence. You can monitor this at the quantitative way. You can also use filters and also to discriminate actually that the change in color, there's not only an increase in luminescence, but also in change in color, the expected shift. There's different ways of doing this. And of course, we confirmed by PCR that there was these plants. Marta Vázquez, checked all the plants and found the level of contamination. So actually, the infection, actually the tomato plants were being infected at the same time that the sentinel plants were reporting this infection. And again, some of those plants we checked that actually there was this shift in mobility due to the break of the bread. So that's it. That's the whole picture. I hope I convince you that autobioluminescence provides inexpensive and high throughput reported for evaluating synthetic gene circuits in plants, not only as sentinels, but it opens in our lab at least, and I think in many other labs, many other possibilities to monitor at different levels, not only at the plant levels. That, well, we managed to engineer bread with this switch, and that at least proteoviruses can be detected in a very specific manner using this system. And then it brings me to the initial question of, can we generate robust, valuable traits using these gene circuits? And the answer is we are not sure. Because, well, I show you that we are there. We are producing plants that you can consider that we are learning how to engineer them, but there are many question marks that we would like to continue exploring. Most engineered lines do not show the expected behavior. I show you this with the copper. So we don't understand why, but only a small fraction, probably there are positional effects and so on. The way we plug these elements into the genome, it matters. So we really need to be able to target the circuits in specific locations of the genome. This will make a difference, and this is something that is still missing in plant biotechnology. The problem with silencing in the long term, and this is something that is not in our case, but it has been widely reported. There's a real problem in the industry. And yeah, we don't know about the robustness in real life, robustness, productive conditions. Is something that is still an open question mark. But we are planning to do new experiments, more circuits. We have many circuits and also in the making. We are now able to produce more multicolor outputs that give us more possibilities for multiplexing. We are into automation. I think automation is important to understand, to improve the cycling, and also, as I said, we need higher insertion technology, and also other plants. And that's all. Thank you for your attention.
Thank you very much, Diego. Thank you very much indeed. I am sure that there will be many, many questions for you. Well, Pablito.
Hi, great talk. So I have several questions.
Okay, I hope I can answer them.
Yeah, of course. Let's go with the toughest one, I guess.
Okay.
Regarding the Sentinel system, that's pretty cool. But when you show the loose system has a 35S P19 module. That is obvious because you have so many 35S stuff that you need to stop silencing. But potiviruses are RNA viruses. So if you put a P19 overexpressing there, you are making the plant more susceptible to viruses. So you are using a Sentinel to detect infection, but the Sentinel itself is more sensitive than any other.
Is that a problem?
Of course, because you are not sure that the other plants will be infected. Because
Well,
this one is very susceptible, so it can be infected.
Yeah,
doesn't mean that the other ones are
From our
infected.
point of view, this can be. In the stable plants, there's no P19, right? In any case, there are 35S. You can change the promoter if this is a problem. But actually, one of the things we like, maybe we're wrong, but one of the things we like about using Nicotiana ventamiana as a sentinel is because it's apparently has more susceptibility to gene silence, or it's less susceptible to gene silencing, because you know that they have this mutant RNA dependent RNase polymerase is more sensitive to any kind of viruses. So as a sentinel, not as a production crop plant, but as a sentinel for detecting the presence of the virus and to be an early detector, I don't think susceptibility is a problem, because what you want is just the opposite. You want to make sure an early warning, and if you want an early warning, susceptibility is a pro, it's not a con. That's my point of view, right? Although, definitely, if this is a problem, then I mean, you need to go to version 1.20.30, and definitely, I'm sure that won't be the final version. But I think in this case, this is a pro, and the fact that we like Bentamiana for many reasons. But one of the reasons is the susceptibility due to viral infections, right? Actually, it's the reason why many people use Nicotina Bentamiana as a tool, right? I don't know if I convince you.
Aphids coming into the crop. So it's good to have a more susceptible plant.
Yeah, a different thing is that, well, if Bentamiana would be susceptible to the same aphid transmission as the others. There are many options. I mean, this is not a final program, fortunately for us. You could think of the same species also as for certain reasons, if you can make sentinels of the same species, probably would be more reliable. But in this case, at least for these experiments, I think Bentamiana it's quite nice. In the case of aphids, probably you will have to see to what extent also is transmissible, not only by viral inoculation, but also with the aphid, right? That's a point.
Anybody can steal my microphone. So for the startup point of view, so you're using Benti, why not use another forage that produce more leaves, or even, I don't know, cauliflower that produce a lot of tissue?
Well, for the time being, we're using transient expression in the spin-off company. The speed, susceptibility, it's the standard of the industry. I don't see why you cannot use other plants. But remember that we are not that susceptibility again is one of the pros of Nicotina Bentamiana. And Nicotina Bentamiana, if you use viral system, for instance, TMV, Gemini virus, and you try to transfer even to tobacco, I mean, it does an agroinfiltration. There are a lot of resistance. There's a lot of silencing.
I was not thinking about the sentinel, but about the
protein
production.
Yeah, it's exactly the same.
You have like a huge plant, it will produce more, right?
Yeah, but
like
you can grow. Yeah, but do you want a huge plant, or you can plant several plants? I mean, the good thing of Bentamiana, it produces tobacco, produce many seeds, so you don't have a problem of productions of seed production. So higher biomass is true. You can produce more leaves, but instead you can to plant more plants and have the same effect. But the levels of expression, and Sylvester Marionette, one of I don't know if you heard about Sylvester Marionette. Golden Gate, if any of you use Golden Gate, was invented by Sylvester, one of the most intelligent person in this work, in this field that I've ever met. And Sylvester, who invented Golden Gate, calls Nicotina Bentamiana the plant of God, because it's amazing, but probably for this defective RNA-dependent RNA polymerase or whatever else reason, it turns out that most of the systems we are using were much better and more productive at least for agroinfiltration in Nicotina Bentamiana than any other leafy or whatever plant, right? So that's why it's the standard of the industry, and we can discuss about the reason for that. There are many different hypotheses for that, and then these feedbacks, because there are more tools developed for Bentamiana, so that at the end is like with CHO or with other systems, that at the end is the pioneer advantage, that at the end all the tools are produced or designed for this specific chassis or species. So that's why. But I'm ready to. I'm very open to any other production system. Huh? It's not. I'm not. I'm quite agnostic about this.
I have more.
Sure.
Anybody? Cube. So once you activate Cube with Cooper, you can turn it off, or once it's on,
No.
stay off.
We are working in systems that are reversible. This is not reversible. This is irreversible. So that's why we like. So the goal of this, particularly with the post-harvest production, the nice thing is that you don't need to revert it, right? So you can. So you can switch it on at the final stage, and then it produces, and then you throw out the plant, right? That's the point. Reversible systems are more difficult. We have a paper years ago on a reversible system use recombinases, but in order to do this, you need to have a reversible factor. Certain recombinases, tyrosine recombinases, I think, this is, I don't remember. So now this is recorded. So if I say something, I say something wrong, everybody will. So you can add a reversible factor. So we have a system also developed in the Goldberg system in which you have a promoter flanked by recombination sites, right? And you can use this depending on the orientation of the recombination sites. You can use a recombinase to invert the promoter. So this is you can switch on in having the promoter in the direction of the CDS, and you can switch off if you turn it in the other way around in the presence of the recombinase. So the recombinase, but there are certain recombinases that if you add the recombinase plus a reversal factor, you can go in both directions, right? So in that case, you can do a reversible system, but this is not, and this is quite difficult to work with. The reversibility, it depends on many factors, and they have to be balance of expressions. So the reversibility with the recombinases is very tricky.
So thank you for the talk. Very nice. I knew some of this stuff, of course, but the Sentinel part, I think, is really, really nice because with this intercrop strategy, you don't have to make transgenic, to detect the tomato transgenic or whatever. So I think it's really cool. But my point is, I know, with viruses you have enough, but are you planning on doing something with other types of biotic stresses, bacteria or fungi? You planning on that?
Yeah, it turns out that there are many things. The nice thing about this system, I mean, you can think of many different systems. We like very much the protease stuff. And there are many things that happen in the cell mediated by proteases and specific proteases. So we are focusing, in the future, we would like to be able to detect other factors or other stress factors also based on the presence of specific proteases. This could be a very nice, and it could open the rainbow of different detection systems that you can work. Yeah, you're right. Regarding this, if you notice the two examples that we decide to develop are those that we think are, I mean, we are talking about transgenic plants, so this is always difficult, right? But we are trying to work with things, with strategies or with approaches that could be, you can see on the horizon of applicability. In bioproduction is right away, because in contained conditions you can work with it. So if everything that we develop in biocontained in Ventamiana and for bioproduction, it can go directly to industry if it works well, and it works in the real work, but it can bypass any regulation, because contained conditions you can produce it as many transgenic plants as you like. And the other part is, as you mentioned, is more difficult for regulators to accept, but it's not the transgenic plant itself, is not the production plant, but is the cropping. So what we think about this, we always try to think in a certain way to final applications. And of course, transgenic plants for food are not in the scope now.
Thank you.
By the way, these are tomatoes, huh? This is the next step. Those are cool. They're very cool. Yeah.
Thank you very much for the talk. Very nice. I go back to the usability of the Sentinel. I find that from the synthetic biology point of view, and the circuit is amazing, but I guess you need to design a Sentinel plant for each pathogen. even for each specific Gemini virus strain. So how applicable is this from the agricultural point of view, and how much do you need that if you already have roses for oidium and other plants for other things? And how much would that cost to the producer that would have to buy to a company that produce each of these plants? I guess you need to have a ratio of sentinel plants for production plants, so it would cost a lot to the agriculturers. So,
well, they are very good questions, actually. They are very good questions. We thought about this. Well, I mean, there's a trade-off between the specificity and the range. So for certain applications, probably it would be nice to have, as you said, a broader sentinel, a broader scope sentinel. We're thinking about this, and we are trying to develop something that is related with silencing responses in the plant, something that could also give a... So I think both things could be interesting for the farmer. That's my idea. I mean, it depends which is the stress, or, for instance, if there are certain situations in which what you really want is, you know, the virus you don't want to have in your culture. I mean, there are emerging diseases, there are, and you want to be specific about this detection, not a broad detection. I think in these cases it could be useful. There will be other situations in which you only need to know that there's a virus there in order to, or a bacteria or something like this, right? So I only can say, please regard this as an initial approach in which we are trying to develop the basic of the circuits, and I think we would be able, if this works, I think you could develop to a broader, to increase with other strategies, the scope in the cases it's necessary, and to narrow it in other cases. That's my point.
Hi, very nice talk. I have just a curiosity. What about roots? Are they also luminescence, or?
Well, it depends, of course. In the case of the viral infection, yes, we also saw movement through the roots in the case of viruses. In the case of constitutive expression as well. Also, we have tomatoes sometimes that you put it in the pot in order to make the picture, and you see here there are a small root that is very brilliant. The roots are very interesting. Actually, we have a project now to use hairy roots and to use root cultures also as a sensors for environmental stresses based on bioluminescence. Because one of the nice things about roots is that there's no how do you call this? There's not fainting because pigments are also quenching the luminescence, and in the case of roots, it's very nice because you see sometimes even more intense than in other parts of the plant. So, and I think roots are very interesting also for early detection of many kind of stresses. So, and actually we are, just as we speak, starting a project on roots specifically.
Thank you.
And they are very nice because you can take pictures without any light contamination. So roots are, I like it very much, and it's one of the things we're looking into it, and others as well, right?
Thank you very much. Is there any other question in the audience? If not, thank you very much again, Diego. Un abrazo.
By the water neath the Mexican sky, drink some margaritas by a string of blue lights, listen to the mariachi play at midnight. Are you with me?